we are. And we had a snow day on Tuesday. USM opened at 10, and my understanding of USM policy is that if USM opens after class has officially started, well, that class can't meet. So that's where we're at. And I showed these internship opportunities through the Career Center on this snow day lecture I made. And so what should we do this week? Okay, so when there's a snow day or other interruption like that, I just make a class meeting with nobody in the class. And so I did that, I posted that. And so what should you do this week? Well, in that lecture recording I made on Tuesday, there's a drawing assignment that we would have done in class, and instead of doing it in class, you're required to do it at home, and send me email pics of those by next Tuesday. And in the Brightspace announcement where I put the link for that, I said, I make those as if we're just rolling on, you know? And so don't wait and watch it next Tuesday when the assignment is due. Rather, watch it as soon as you can and get that done, because I'm going to talk today as if we had class, and there was the recording, and we're moving on from those topics like that, okay? So that's the first thing. Then the next thing is set my alarm for 10:30. So we'll have a quiz 2 at the end of class today. It was scheduled to be on Tuesday. We'll have it today. And when you hand in quiz 2 as you leave, you can pick up quiz 1. Okay, like that. And so that'll be out up here as you hand in quiz 2. And then, we're gonna have that quiz. Then we're pretty much done talking about what I want to say in chapter 1, but you can still ask questions, and you should, you know. And so if you have questions about chapter 1, those can come up at any time. We're going to be moving on to chapter 7. And that's the last chapter that I can put on electronic reserve. So everybody has access to these first 2 chapters. Everybody can keep up with the course through then. And after chapter 7, we're going to section 4 of our book, chapter 16, like that, okay? And chapter 7 is about transcription regulation. So it's really the core of it is, well, look at the muscle differentiation. And muscle differentiation is just a universal example of changing gene expression in a eukaryotic cell, okay? And then the next one is this. So when I grade quizzes or exams, what I do, and have done for several years, bunch of years, is I almost never write down comments. I just mark points off, and then I take notes, you know, like off to the side. And after I'm done grading, what I do is I make a video of myself taking the quiz or exam, and I call that the answer key video. And I say the correct answers, and I anonymously share some of the wrong answers. And I do that for a bunch of reasons. One, the most important ones is there are often situations where several students get the answer wrong the same way. You know, so there was a common misunderstanding, right? And I should tell everybody about that, right? Because if some students get it wrong because of that reason, maybe everybody kind of has a little misunderstanding about it like that, right? So then here's another main reasons I do it. Lots of the time the answers are fine and they get full credit. On quiz 1, most students got them all right, you know, and others got a few wrong like that. But really often I have to give full credit. It's a good answer, but there's a better answer. And so I want to share that, and of course I want to share that with everybody. And so when I make an answer key video, I think, oh, this is important for everybody, even students that saw the full, got all the points, right? Because there's more learning to do. And so I've done that for so long that I have no problem putting new content into the answer key video. Another example, right, where I'm like, oh, so students didn't quite get this, I should explain it better. I can explain it right now in the answer key video, like that. You know, in other cases, right, the instructor comes back in and he says, you know, I already missed this one thing on the exam. I want to go back to it and talk about it. So I just do that more and more in the answer key video. So it's required. And to help you get on board with that, we have an assignment. And the assignment is, at the end of class on Tuesday, we're just going to take quiz 1 again. And I'll grade it, but with more expectation that, oh, you saw their answers. That is, I'm doing that to get everybody on board with the purpose for that. You know, make sure you understand it. And then also, it's a difficult thing to talk about, but there's always a really direct answer like that. And it's good to know that. I have to use a lot of words and examples to explain things, you know, to help you get it for the first time. But it's good to, you know, kind of drive toward that answer. And in that answer, molecular biologists are really specific, fussy people, you know? Like if we're talking about the engine of a car and you say, yeah, I probably need to talk, you know, talk about the tires a little bit, I'm like, nope, 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 you know, like that. And I am very, very fair, and you know, I'm trying to give you as many points as I can, and I'm not grading you on grammar, but I'm hoping, you know, to demonstrate that and have us try to drive toward that goal. Then another thing about it is our exams, we're preparing for those, and our exams are 50 questions. And I have given that type of exam before. You can get it done in class, but you're just gonna have to stay focused, and it'll fill up the time. And in doing that, we don't have time for you to add like extra stuff. Here's a quick example. Sometimes students answer questions by repeating part of the question. You don't need to do that. Then sometimes students will just do something like this, where I said one of the questions was, you know, on the first quiz was did Darwin use phylogenetic or phonetic approaches? And you could just circle it, because I'm not asking you to explain it or provide examples. So it's okay for you to be like, okay, you know, he's not asking me to explain this, describe it. I might say, describe it briefly, explain it briefly, right? You know, the wording of the question matters, and you don't have to get anxious about that, but just be aware, and I'll mention this again, that there is an issue on the exam that if you're like, you know, not moving through it, you'll end up running out of time. You know, you gotta kind of move through it, but not, oh God, I gotta run a marathon. Just the idea of let's just move through it. So that's our assignment for the answer key video. As we're just gonna take the quiz again at the end of class on Tuesday, like that. And we don't do that. I've never done it before. This is the first time I've ever done it. I've had other kind of assignments, you know, like that. And we might have an assignment after the first exam, like maybe go through the answer key video and correct, you know, some of the ones you got wrong, something like that. But this first time I just said, we'll just start the course with it this way, so you can see it and see what I'm trying to do every time with answer key video, okay? So that's that one. And then we have a homework assignment. It's a video that I'll post, and I just call it the Wisconsin and Boston. I've mentioned it before. Students struggle with this issue that comes up in this course, and our author demonstrates it physically in his life when he was your age. He was in Wisconsin, he studied things one way, and then he got a job in Boston, and he totally left the Wisconsin approach behind. So it's about that, but in doing that, not just, you know, limited to that, it's explaining a whole bunch of stuff in that context, like that. Trying to have it be helpful learning, because it really helps not to know the name, or know the years, or know locations, but just to have some examples where you can spread out your learning like that, and like that. Like the lab in Wisconsin, you know, I always think of the lab and the professor, because he looks like my dad, who did the work, you know, and that's very helpful to organize all your learning around, oh, my dad did that work, my dad didn't, but you know, and so I'm not gonna ask you about Wisconsin and Boston on the exam. I'm just trying to help you set it up in your own mind all the time, thinking about the physicality of this and the way of spreading it out, okay? So there's that electronic reserve thing. There's our flow, so we're gonna be, you know, moving through here and on to here, but we can always go back to that, and then we're gonna go to there, cell signaling like that. So I should just pause and see how people are doing, and if anybody has any questions, and it could be anything. It could be about chapter 1, could be the quiz today. Yep?
I just had a quick question. Is there any way to know a little bit ahead of time if a specific chapter will have attached to it like a homework assignment or drawing assignment or a quiz so that for time management purposes?
I would say no.
Okay.
Because I'd say that the assignment, so I'd say this, that when I am using a textbook, I am trying to tell things differently than in the textbook. And so you have like two people explaining stuff to you instead of one. And so a lot of times my assignments are kind of arcing across multiple chapters like that. And here's a good example of that. So on the very first day of class, I showed this FOXO. And FOXO shows up here. And I've said, our course has a big picture, and it has 3 big pictures. And we're running on one of them, and that's how do you turn a transcription factor on and off? What are some examples of that? The second one is, the big picture is, how do cells receive signals, like hormone signaling, and respond to them like that? And so right at the beginning of the course, you're not leaning toward that, like that. And so it's hard to say, oh, this is an assignment from that chapter. What is good to think about is we'll say the names of the numbers of the chapter, but it's better to think of the topics, because the topics can be simplified like that. And so we could be paying attention and saying, I see we're heading towards cell signaling, and we are foreshadowing it there and there and there and there, like that. Yep. Yeah. Any other questions about anything at all? Always remember. Yep.
So the questions that you have on the quizzes, will we see those again on the exam?
Yeah, so that's a good question, and here's a way to frame it. So we're going to have 3 exams and a final, and the final exam is going to be cumulative. Will some of the questions from the first 3 exams be on the final? And at first you'd say, well, I would say, well, no, because I try hard, to ask you questions that are important, and I don't have enough numbers, I gotta ask some more questions like that. But then on the other hand, I also make these answer key videos, and I take time to do that. And so that's something I want you to learn, right? I ask questions that are important, and if you got them wrong, I want us to follow them up and learn them. And so on the other hand, I kind of like the attitude of putting some questions just directly or similar, on an exam. And so the answer about quiz 1 is, well, I shouldn't need to, like that, and I don't really have time to, but on the other hand, I want everybody, we're doing it twice, right? We're taking time in class to do it, right? And I also, these are, the start of our course, these are really foundational. So it's, I describe them as a prerequisite like that, right? Because we have a requirement of intro bio, genetics, chemistry in here like that, So I'm like, we gotta get going, yeah. So, yeah, like that. So maybe, like that, yeah. But, why and why not, those are the reasons like that, yeah? Other questions people have? And remember, I bring my book, you could just point to somewhere in the book and say, what's that, right? And so, you don't have to be fancy in your framing of the question. You can just be like, I don't get what's that point, like that. Because there's a lot of stuff in the book that we won't normally talk about in class, but we can, And what I'm thinking about is, I look at the book and I'm like, well, the author does a good job explaining that. Hopefully the students can understand that, and if they don't, they'll bring questions to class, like that. Yep. Other questions people have? Okay, you can interrupt anytime. It's my job to fit your question into, class. You don't have to worry, like, we're talking about something different, just raise your hand and blurt it out. Don't worry about it. Yep. All right. Okay, so that's our flow, like that. And so moving on from chapter 1, I've already mentioned we'll never read chapter 2. We've got prerequisites for chemistry in our course. There's a couple of times I'll use a figure from chapter 2, but you don't have to look at chapter 2. We'll describe that in class. So just to give you a feeling for why we're skipping some chapters. We're skipping some chapters because we're rolling toward this section 4, which is the heart of our course, so let's get there. And after we finish those chapters, we'll loop back and cover some additional chapters, but not chapter 1. You're welcome to look at it, right? The author put that in the book, trying to help you, do well, but you definitely don't need to. If I put a question on the exam, like, hey, this is from chapter 2, I'll be like, no, it's from intro chem, like that, right? Yep. Then I also mentioned again, I mentioned this before, chapter 4 is an introduction to molecular biology. So we saw a little taste of cell biology in the first chapter and really very little molecular biology. We might never read this chapter, maybe we'll have time to, but you can definitely look at it and you can always ask questions because you are welcome to ask questions from anywhere else in the book, any other book, any website, and definitely previous courses you've taken. Because some of this stuff you've heard some of before, like that, right? And I'm a super pleasant person, but I'm also a molecular biologist, and so I'm like, yeah, nope, this is the way. This is the way, right? Like that, yeah? So just that, and I won't, mention that again. And then I want to point this out. In molecular biology, there's a feeling to it, an approach to it, a philosophy, a strategy to it, and it's this flow, like that. So we're learning a lot of nuts and bolts details because that's what a molecular biologist does. He says, oh, you have a bike? Well, we're just going to take the bike apart and we're going to learn about every part in detail. And then we'll understand the bike. Yeah? So it's a certain strategy, that arose in science, in biology, 70 years ago, and it's just very, very successful. And it's like, let's put a little tiny brick in one at a time. But let's make sure it's a good brick and the building's not going to fall down like that. It's just really good. So one of the things that I've always thought was great, but it also helps teach the topics, is this. Discoveries get turned into tools. So in that snow day lecture, one of the parts of the drawing assignment is to explain how you can make a mouse, a transgenic mouse, that has a transgene that would cause muscles to glow. And that's a good way to deepen your understanding of the organization of a gene. A gene is made of 2 parts. In a test tube, we can just mix and match those from any species on our planet because the DNA sequence is the same story, same biochemistry like that. So we can put a jellyfish gene into a mouse or a gene from any other organism on our planet, and the mouse will deal with that, like that. So that becomes like a good thing to explain on an exam. How would you build a mouse that has muscles that glow? Like that, yep. So a discovery that gets turned into a tool, like that. So here's a second example of this idea of that some discovery gets turned into a tool. And I actually describe it in that Wisconsin to Boston homework assignment that you'll be working on. And it just comes up. And what it's like is this, that the scientists so I circled it there, it comes up there. So I won't explain it in detail here, but just to show you, it's another example. And we're using these discovery examples that get turned into tools for discovery as ways of helping to teach the topic. And so again, another example. That's the guy who looks like my dad. And yeah, he's a cool guy. So anyway, he's running a lab in Wisconsin, and he's studying retroviruses. What's up with retroviruses? So there are 7 major classes of viruses on our planet, and one class is retroviruses, and that's what he's working on. And retroviruses are RNA viruses, and they have to copy their RNA into DNA when they infect the cell. So they have to have a gene that codes for an enzyme that can do that, and it's called reverse transcriptase. And so this guy, my dad, won a Nobel Prize for studying just retroviruses. And it was for really 2 reasons. And one of the reasons was that became a powerful tool that's used in experimental labs, that reverse transcriptase. So for example, in the classroom lab in the afternoon, we're using that enzyme next week, like that, for our experiments, like that. So it's an example of that, and it comes from this. So it comes from another drawing, and in the homework assignment is in Wisconsin, They're just working on the replication cycle of a retrovirus. The normal replication cycle of any of these retroviruses. HIV, AIDS virus is a retrovirus. This is how it gets replicated. And the original AIDS drugs were all targeting that reverse transcriptase. We don't need that, so let's try to crush that thing and slow it down, right, like that. So you could have to draw that, on an exam, like that, right, okay? And that's happening in Wisconsin, and there are really two things about we need to know that cycle, but they're really focusing on two things. And one of them is the normal retrovirus replication cycle includes that retrovirus, our reverse transcriptase rather. So it's infecting with an RNA, it's going to get copied, and it first gets copied into DNA. And so we want to learn that, and partly because it's a cool thing that's used, lots in research labs, but partly just because that's the natural process of this retrovirus, like that, right? And molecular biologists, a lot of molecular biologists are virologists. So I said, there are 7 classes of viruses on our planet. That's a great molecular biologist named David Baltimore that said that. There are 7 classes, and I could start crying again. And it's too bad that I break up, choke up easily, but this is my super passion in my life, so I'm just crying about it all the time. And it's described some in the Wisconsin Boston video. I just am showing you that, we want to pay attention to that and just kind of repeating it or kind of foreshadowing. You want to pay attention to that, right? So it's used in reverse transcriptase is used, okay? It's just that enzyme, and then it gets used in lab, and what it does is it copies RNA into DNA. Then the second thing that we want to pay attention to in that retrovirus life cycle is the second thing that comes up in Wisconsin, and that is to go through the replication cycle. After it copies the RNA into DNA, it inserts that DNA into the host genome, into a host chromosome, and it inserts it randomly. And then the host RNA polymerase starts copying it, like if it's transcribing a gene, but it's actually transcribing the retrovirus, and we're getting RNA, and that RNA gets packaged and leaves the cell. But that concept is a key one, is it inserts, and it inserts randomly. Because what happens is, once in a while, a retrovirus could insert next to a host gene that is involved in some kind of growth regulation maybe, and it could actually pick that up. And that's exactly what happened in several retroviruses, is the retrovirus during its replication cycle accidentally got a host gene. How to do that? Because its natural cycle involves getting next to host genes, it could mess that up. And viruses are really sloppy because they make lots and lots of copies and they do it very sloppy. And so that's shown here, and this is then the final chapter of our book. So, back to Violetta's question, we're setting it up like that. And this just shows the normal, a normal genome for a retrovirus, and it has the polymerase gene like that, retrovirus, RNA, reverse transcriptase. And then here's one of these mutants, and it's picked up a host gene. And I've already talked about, the author studied this in Wisconsin, and he found a host gene called RAS. And other people worked on it too, but our author found it in that lab. And that's the other reason why that guy that looks like my dad won a Nobel Prize, is those two things were a big, big deal. And people are finding it elsewhere, but they were rocking on it in Wisconsin. And that's why our author, at your age, our author went to Wisconsin. He went there from Miami, Florida, where it's really nice, to Wisconsin, where they have wicked winters, for a reason, like that, yep. And when he moved from Wisconsin to Boston, he left the retroviruses behind in that lab. He went to study RAS, but no longer in a virus, now in a mouse, because that's where the gene came from. And he found in Boston, nothing to do with viruses, there are mice that have cancer that have mutations in RAS. Yep, so that's a thing, and that's that guy. And again, we're not dwelling on him, but that's his thing. And so here's just a table, not in our book, that shows some of these retroviruses that actually picked up a host gene out in nature, and the host gene became defective in one way or the other, we'll learn about. And those were, we're going to call those oncogenic retroviruses. Because that's what they are, cancer-causing retroviruses. And that will help you appreciate these are mutant viruses. The normal wild-type virus does not cause cancer. These are ones, like strains, versions of the virus that accidentally picked it up and caused cancer when they infected further cells. And in 1970, scientists studied about 20 of those, about a couple dozen of those, and they came from not humans, but a variety of other organisms. All of the examples were genes like RAS and SARC and so forth that have orthologs in us, but they didn't start studying them in us. They started with a virus that was found in a cat, one in a chicken, a turkey, a monkey, a mouse, a couple in rats, other birds like that. And they were just these curious viruses that scientists had found over the years that would just cause cancer as soon as they infected a host. If you get that virus, you're getting cancer now. And it would cause specific kind of cancers. So one of them is called Maloney-Murine leukemia virus, because it was discovered by this guy Maloney, and murine means mouse, and leukemia means a kind of cancer, like that, right? And we'll call them oncogenic retroviruses. A lot of time in our book, it just says a retrovirus, retrovirus like that. And so we're going to distinguish between just the normal retrovirus, and we'll soon leave those behind, and the oncogenic retrovirus that has the defective host gene. And we'll soon leave the oncogenic retroviruses behind and just talk about the host genes, okay? And I mentioned last time, they really typically call these genes by three-letter names. And so RAS is one. And we'll meet some of these other ones in the fourth section of our book. And we'll meet June and FOSS and MIC and ERB and AKT, like that. So we'll meet 6 or so of these, and they are in our body, and they're doing normal functions in our body right now, and each of them could get mutated and cause cancer. Cancer is a multi-hit process, multi-mutations, so maybe 2 of these get mutated, and we'll talk about examples of that. But some of these are really dangerous if they're under control. And in the oncogenic retrovirus, it's just one gene that just boom, that organism has cancer. So it becomes a story of like an Achilles heel that will look at pathways normally happening in our body, and there'll be a vulnerable point, a vulnerable step. And RAS is a vulnerable step. It's got a job to do, and the job RAS does is it's on pathways where a cell is going to be stimulated to grow. And there are mutations in RAS that could cause that RAS to just go, like that. Without being told, it just go. Okay? So a little of that, and again, that's covered in that Wisconsin and Boston thing in more detail, but it helps kind of link that importance. So here's from a future chapter, probably definitely chapter 16, here's just the RAS signaling pathway. So where does RAS fit in? And it only fits in one place in any animal, anytime. And RAS is here, and what we have is we're looking at a cell, and there's a receptor, and the receptor is going to respond to a growth factor. So the growth factor is going to be a molecule like, like something like a hormone or another kind of signal that's just out in the extracellular environment, binds to a receptor, and then this receptor doesn't bring that signal into the cell. Instead, the receptor gets activated, and it triggers a biochemical pathway into the cell, and that cell makes a cellular response. So we call that cell the target cell, and some other cell made that signaling molecule, that hormone or whatever it is. And so there's a source of a signal somewhere else. It's floating around as a molecule, binds to the specific receptor, and that causes the target cell to respond. And that's chapter 16. It's all about cell signaling. And that's a chapter I mentioned before in intro bio. In Campbell, it's chapter 11, and I'm like, oh man, that's a pivotal chapter in our topic, in our course, and there's no doubt about it. But if a person's a microbiologist and they're teaching intro bio, they might not cover it because there's not too much cell signaling in microbiology. They're a single-celled organism. But we're talking about multicellular animals coordinating plants, like that, right? Okay? And so this is the pathway, and RAS fits there. And then it's a series of steps like that. And we'll talk about in chapter 16, well, why are there so many steps? And there's some general reasons for that. But RAS fits here, and for us right now, what happens is the signaling pathway leads to a protein called ERK. And ERK is a transcription factor, and when ERK gets phosphorylated, it activates ERK, and it turns genes on. So in this case, and in every case, RAS functions by changing gene expression in the target cell, always. When it's mutated, maybe we don't need the signal anymore, and the target cell just says, go, go, grow, grow, grow. And that's how RAS causes trouble. Can get mutated and cause trouble. And we'll learn about that in detail. But we'll add, I said ERK, I should have said ELK, sorry. So ERK gets phosphorylated. We'll learn about ERK, but ERK actually phosphorylates ELK. Sorry, apologize. ELK is the transcription factor here, and ELK gets phosphorylated and it activates the gene, okay? So it goes RAS, RAF, MEK, ERK, ELK, like that. And we'll learn about that pathway because there are other oncogenes on that pathway that we'll learn about. And so, also just to mention this, our author likes to make these little sunbursts around oncogenic proteins. So it says RAF is an oncogene and makes an oncogenic protein like that. So we're leading into that in our course. It's a normal signaling pathway that's being used all the time in our bodies normally and can get out of whack. So it activates ELK by phosphorylation. So remember, we're building on this big picture of you got transcription factors, and they're going to regulate gene expression. And they're going to get turned on or off. They're the big story in the first step of gene expression because they decide, are we going to transcribe this gene into RNA or not? So one of the reasons that we only make insulin in the pancreas is because the pancreas cells have transcription factor that activate the gene. There are other reasons, but that's kind of the first one to say, like that. Muscles. Muscle proteins are there in muscles because the RNA is there. And if you dial it back to the muscle precursor myoblast, the protein's not there because the RNA's not there because the gene didn't get transcribed because the transcription factor's not on. And we started that out with our course with MyoD and myogenin. They're pretty simple. When they get synthesized, they get to work. And we've already started talking a little bit about other examples. FOXO gets synthesized, but it's not always active. If FOXO is phosphorylated, it's turned off. It's excluded. When ELK is phosphorylated, it's turned on. So phosphorylation can turn a protein on or a molecule on or off. It's just got to figure it out each time. So ELK gets added to that. Phosphorylation by a kinase. Kinases are enzymes that add phosphates to target. And we've got two examples. And they're kind of interesting and significant because one turned off, the other turned on. And will we have to answer that on the exam? Oh, yeah. Yeah. In chapter 16, we learn about that signaling pathway in detail, receptor and all those steps along that pathway, because that's really what this course is about, because the course starts out saying, DNA is pretty interesting, and then, DNA is organized into genes, and genes get copied into RNA, and those get activated by transcription factors, and transcription factors are not always on. They get turned on and off, and that creates a lot of the orchestration of life on Earth. Every cell on our planet always has DNA, and almost always has DNA. It's to say, a rare exception, human red blood cells have thrown away their DNA. But in general, DNA is in our cells, and all the cells on our planet that have DNA are not using it all at the same time. They're using portions of it, and as a first step story, transcription factors are a big deal. So how do you turn them on and off? And it turns out, it's really common in stuff like people, that it's going to be some extracellular signal that's going to be regulating gene expression in just this way. So we now know the specific, and we'll know the specifics of this in a few examples in chapter 16 and on, as models, as examples of, this is interesting to see, how these are regulated, like that. We've already started talking about our next one, and I've talked about that little class and in some assignments, and it's this estrogen receptor, because the estrogen receptor is a transcription factor, and it gets turned on by estrogen. So it's a hormone-dependent transcription factor, and hormones, molecular biologists, biochemists call those ligands. So it's a ligand-dependent transcription factor. That's fascinating, because at the time of puberty, that hormone can activate different gene expression in a whole bunch of our cells, right? We have a lot of changes in our bodies during puberty, and one hormone does it through activation of gene expression. It's a transcription factor. And that's a satisfying example of how the genes are off till they are needed, and our transcription factor is going to be the first step in activating. And so, like that, okay? So, in this RAS pathway, sometimes we'll talk about normal wild-type RAS, and sometimes we'll talk about mutant oncogenic RAS. And we just want to make sure, to say, this is just a normal one, like that, okay?
What do you mean by wild type?
Yeah, so wild type is what's probably present in all of us. And so I'd say wild type alleles. There are a bunch of different versions of RAS that are slightly different, but they're all normal. They're all doing kind of their regular job. Something that you'll never see, I'll just draw this out, and so you'll never see this because it's a hypothetical drawing, kind of like that, but it's a nice visual to see and just recognize that, nobody ever does this, talks this way quite, but we can. I gotta get this thing going for some reason. Is that not turning on? Sorry, my little drawing thing is missing out. I've just kind of lost my Bluetooth connection with my little drawing pad, and so that just means I have to pause here for a moment and go back to this and go to this. All right, and we're still recording. All right, so let me just draw this, okay? So we're talking about any gene, and we're going to talk about the alleles of a gene. And we could have we could just talk about it and think about it like this, that it's going to be a kind of chart. And we could say, this is wild type. And they call it wild type because it's like, go catch one. It's probably going to be wild type. And there could be a variety of alleles that are wild type, but they're all working pretty well. And this chart is going to be just a range of how well is the allele working. And it's almost impossible to put, like, this is just working at 100%, doing a great job. But that's what we mean, like that, is it's just working fine. And it would be really hard to quantify the activity of gene like that. So that's why you don't see it. You don't see it. But if you look at this chart, it's a satisfying way to see some genetics. And that is, a mutant allele would take us away from the wild type. And they're random mutation, so they may typically go down. And they go down to a specific amount that, again, you wouldn't ever have a number for, But an allele functions like that, where it's like, it's working not as good, but like 87%, 62%, like that, or 0%. Geneticists love that because it's no function, it's a null allele, like that. And there's a name for these, they're called loss of function, which is kind of imperfect because it doesn't mean a full loss of function, it just means allele that has lost some function. And then there's another term, and we're learning those terms now, they're called hypomorphic alleles. And then there's another term we'll learn because they don't put numbers on them, but geneticists like to study genes by using a bunch of different alleles. And so, because it's helpful in different genetic experiments, and so there's a set of alleles here that's just vaguely listed as like that. You can decrease it some, but not a lot. And they're called, believe it or not, weak alleles. And then there's a set that are down here that include the null allele, and those are called strong alleles. And geneticists talk about those that way. And let's just talk about a specific example. So let's say that we were looking at that myogenin gene, and we didn't know about it. We were geneticists, 50 years ago, and we're looking for mutations that don't make muscles. And we'd love it if we found one that made no muscles, null allele. We'd get a lot of work on it. But a geneticist also likes to study strong alleles and weak alleles. Why? Because if you think about it, a strong allele of myogenin means that animal would be dead. If it was a mammal, it would be spontaneously aborted early. There would be no, fetal heartbeat, because it's just got a big problem making muscles. Probably doesn't have any. A weak allele becomes interesting to a geneticist because it might be alive, right? It might survive and have problems, like, problems that you would have with weak muscles, like that. And that's cool. And here's one reason it's cool is because the geneticist first discovers this gene we now know as myogenin, for example, and then they say, let's find more. And what they'll do is they'll do a second round of searching, they call it a genetic screen, for mutations. And what they'll do is they'll start out with weak alleles, because that one already has a problem. It doesn't take it much to kill it. So they could find start with a weak allele of myogenin, and they could actually find a weak allele of MyoD. And they could study them separately, because separately they both survive, but together the animal dies, like that, And so that's why. Why would anybody talk about a weak and a strong allele? Because it's a geneticist, and geneticists are always looking at the phenotype. They'll do some breeding experiment, they'll do some crosses between different strains that have different versions of alleles, and then they'll look at the health, the phenotype of the outcome. So that's where those terms come from. Then, just to finish this off, it's unusual, but there are also mutations that go this way, have an increased activity. And so, they may go up to 110%, like that. So imagine like hemoglobin, and somehow hemoglobin that works better, like that. And there are names for these that we're learning right now, and they're called gain of function. Or they're called hypermorphs. And you start out with, you think, I get this. Gain of function would be the hemoglobin binds better, binds or releases oxygen better, doing a better job. But that's just one kind of mutation a gene can have. So I said, and I don't know how far we've gotten into this in a video, but in one of the video assignments I said, and I think it's the Snow Day lecture, where we're doing these drawings for the assignment, and in that context I say, check this out. You can only have evolution if there's a mutation, a change in an allele, a change in a gene. And on our planet, there are only 4 ways you can change a gene. And one way is you can change the product, change the hemoglobin that's made. And it could be better. You could make a better hemoglobin. right? How could it be better? Well, it might be of selective advantage if it was an animal like a seal, you know, that's evolving to go back and dive in the ocean and maybe needs to hang on to a lot of hemoglobin, right? So, you know, a better hemoglobin could be because the organism is evolving to move into a new location, you know, to spend time in a new location, and it would be better. You know, the animals are wandering around on land are doing fine, but as soon as they start transitioning to try to hold their breath a long time underwater, could be that a mutation in the product would be beneficial and have a natural selection advantage, okay? So that would be where you say, oh, I see, you know, 110% somehow, you know, kind of like that. But again, nobody would talk about that. They just say it's better, like that, right? So then the other three are all in the cis regulatory region, and they are where, when, and how much of the gene gets produced. So that's the cis regulatory region. The mutations can only be in the two parts of the gene because genes only have two parts. They could be in the functional product part, the transcription unit, or in the cis regulatory region. And in the functional product part, there's only one kind of mutation you can have. You could change the product. And in the regulatory region, there are only three ways genes get regulated: where they get expressed, when they get expressed, and how much, okay? Like that. And so a gain of function mutation could be a new location. Here's an example of that. Elephants are mammals like us, and their trunk is their nose, and their trunk has thousands of muscles in it. And 20 million years ago, the ancestor to elephants did not have muscles in their noses. But what do we know? We know that every cell in our body, every cell in our nose has the genes to make muscles. We just never express them. So a gain of function mutation could be suddenly starting to express muscle gene proteins in your nose. And that's pretty much for sure part of the story of elephant trunk evolution. And it arose through mutant alleles that were gain of function. And when an evolutionary biologist comes in and talks, they say these random mutations, they're almost always loss of function. And they rarely contribute to evolutionary change. But those gain of function mutations, they can be pretty cool. And you say, oh yeah, a better hemoglobin, great. And they're like, yeah, or a new location for gene expression, a new time for new gene expression, a new amount. And that's all rooted in the concept of genomic equivalence. We got all the genes in our body, we're just not using them. So what if there was a mutation? You started making muscle protein in your brain. Well, it probably would not be helpful, you know? But it might, you know, like that. So wild type allele, long answer to that. It fits into that. And again, you kind of never see that represented. But a chart is very satisfying to hear what a geneticist is talking about, because they'll say, oh, it's a hypermorph. And you're like, cool, you know? But, you know, we want it, you know, it's physical, it can be represented physically, and you just want to be careful about it, because again, nobody ever is able to talk about that activity quantitatively. But they do have quantitative differences in their activity. It gets complicated because it could be the product that has that quantitative difference, or some aspect of the cis regulatory region. And so there's that. And if you haven't heard that part, the 4 types, it's in, I'm pretty sure, this snow day lecture. Okay, so we want to catch up with that. Okay, other questions people have? Doing great. Okay, all right. So here's this RAS, and look just to see what it does. So it's activated, and RAS is a G protein. So you know, we can draw this out, but I'll just put some of it right here, right now. So RAS is a kind of protein that's called a G protein. And it's called a monomeric G protein because it functions as a monomer. There's just that one RAS protein molecule that's doing the job. So RAS is a monomeric G protein, and it's called a G protein because it's always bound to either GDP or GTP. And that's guanine dinucleotide or dinucleotide phosphate or triphosphate. So it's like ATP, but it's just the G instead. And when it's bound to GDP, it's inactive. And when it's bound to GTP, it's active. So when the growth factor binds to the receptor, just before it binds, RAS is inactive. The growth factor binds, the receptor is activated, and RAS gets activated. How does it get activated? The way it gets activated is it exchanges the GDP for GTP. So these are two different molecules, and it had the GDP, and lets go of it, and it binds GTP. And as long as it's got GTP, it's active. And when it's active, it binds to the next step called RAF, and it activates RAF. Just to mention this, because it's part of this, the RAS has a post-translational modification. It has a little molecule added to it. So the RAS is actually stuck in the cell membrane, and it's floating in the cell membrane. So that's where RAS is always located. It's on the inner side, the cytoplasmic side of the cell membrane, and it can float around, because that's where it needs to do its job. So they call it, it's anchored in the cell membrane. And so it's right where it needs to be to get activated. Then it floats around and bumps into RAF like that. There's a couple other proteins it can activate. And as long as it's bound to GTP, it's active. RAF is an enzyme. It's a kinase. It's going to phosphorylate. So it means like RAF is activated. Boom, 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 boom. And as long as RAS is bound to RAF, RAF is chugging away, activating the next step in the pathway. Eventually, and RAS is going to get inactivated. How does it get inactivated? The way it gets inactivated is RAS is an enzyme and can cleave the GTP to GDP. So when we draw RAS, it's got a place to bind that GTP and GDP right here. And that place is also part of an active site of the enzyme. And so what do we say about RAS? RAS turns itself off. And it's an enzyme, and it just does that pretty randomly after a little while. It gets activated, doing its job, and then it just turns itself off. Okay? All right? It's true, and our book talks about this a little bit, there's another protein that binds to the RAS that really increases the potential activity of the enzyme. But the key thing is that RAS is an enzyme that turns itself off. And if you look, again, 1/3 of all human tumors have a mutation in RAS. And if you look, where is it? It's always in the transcription unit and always in the coding region, and it always causes a change in the protein, a change in the product. It's never a cis-regulatory mutation. It's always the product, and it's always a hypomorphic loss of function. And where is the mutation located? Always in the active site, because it doesn't turn itself off very well. And that turns it into an oncogenic protein, because it just keeps RAF going. Maybe even without the growth factor, which is just like, let's go, let's go, let's go. But it's just not doing a good job turning itself off. An Achilles heel? Yep, because random mutations mostly are going to be loss of function. And this is a protein that has to function to turn itself off. And if it doesn't turn itself off, we just keep the foot on the gas. Yep. And it could be a weak or strong allele, like that. Doesn't turn itself off very well. You might be in some trouble. Might be, null allele, can't really turn itself off, or just keep cranking, like that. Yep. So that's RAS. And is RAS a perfect oncogenic protein to talk about? Yeah, because what you know about biology, you're like, oh my gosh. And every time We talk about the molecular mechanisms of a specific cancer is like that, where you're like, I get that, and that is a huge bummer. A huge random accident that happened in that organism's life to get a mutation in the RAS coding region, in the region that codes for the active site of that enzyme. And don't get confused by the fact that it has this helper protein. The helper protein can also get mutated, because the helper protein helps it turn off. But that helper protein that doesn't happen that often for the helper protein. The big one that we see happening is mutations in RAS. And there are really, I think, just 3 amino acids that we see mutations in RAS, like that. There actually, we have 3 different RAS genes. It's a small gene family, so it could be one or the other, have kind of different roles in different locations. So we'll see that certain very specific mutations of RAS are found in, say, intestinal cancers. They're very common to have RAS mutation, and it's one of those in particular, because one of those 3 versions of RAS is really used normally in the cells in the intestine, and those cells are like our skin cells. You gotta keep replacing those cells. They get rubbed off like that, just like our skin. Yeah. So how about that, right? We're foreshadowing, ahead into the section 4, like that. Okay. So we're looking at that, and then what's one example that we'll talk about this, next week. What's one example of where normal wild-type RAS gets activated in our bodies, and it's under every scab. When a scab falls off, the skin cells are pink. There are a lot of new cells. RAS is involved in wound healing in every animal that gets scabs. So mammals, like that, like that. And if we look back at this, under our scabs, RAS gets activated in those skin cells because there's a molecule under the scab called platelet-derived growth factor. So platelets cause clotting and cause scab formation because they're releasing clotting factors, and they also release this protein called PDGF. And it binds to a receptor on the skin cells, and the skin cells are stimulated. The skin cells on our bodies, I said already, they're always going through the cell cycle slowly. But under a scab, they better speed it up. We're going to heal this, right? And there's a lot of natural selection pressure to do that, right? You want to get that skin back in good shape, right? So you don't end up getting an infection like that when you're climbing around in trees and stuff like that, right? So just classic example. RAS is used in a bunch of different cells in our body, in our chest and skin and lots of different places normally. And that's a good example, and we'll go to that example, and I use that example in my intro bio courses. Here's something I forgot to hand around. It's this sign-up sheet, and I just gave it out. And if I ever do, just tell me. So I'll do that. Then I'm ready to hand out the second quiz, so put your stuff away. And when you get done with this, I'll put your quiz 1 in 2 stacks. A through H will be right on the table, and the K through Z will be over here. So Don Diego's will be over there, and the others will be here. Okay? And so then you've got some homework assignments. We'll just start at the start of class. And there's 2 sides to this quiz. Make sure you do both sides. And look back on this week. Oh, sorry. Yep.